Hello, everyone, and welcome to today's webinar, Targeting the Glyco-immune Checkpoints, Siglecs, to Enhance Antiviral Innate Immune Functions. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit them at genscript.com. Now, we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. Our speakers will answer any questions via the email address you provided at the time of registration. And you may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speaker today, Mohamed Abdel-Mohsen, Associate Professor at the Vaccine and Immunotherapy Center in Immunology, Microenvironment, and Metastasis Program in the Ellen and Ronald Kaplan Cancer Center. Mohamed, welcome to you. You may now begin your presentation.
Thanks for the introduction, and hi everyone. Today I want to give you a quick overview of some of the work we are doing in our lab at the Western Institute. And in our lab, we work in the interface between the fields of in our lab, we work in the interface between the field of virology, immunology. Yeah, not sure why the sliders are not advancing correctly, and glycobiology. And it's pretty obvious why we need to study immunological function during viral infection. But I would like to start with what is glycobiology and why should we care? So I usually like to start with this very simple fact, that is, every single living cell on Earth are covered with a heavy layer of glycan, sugar, on their cell surface. And in the last few years, there is many advances in the field of glycobiology that make it clear that these glycans are biologically active molecules that can impact several immunological functions during many diseases, including cancers. And in fact, there is many important work that's being done on the cancer related to glycobiology in the last few years that you definitely need to check out, but not as much in infectious diseases, including virology. And that's why today I want to give you an example of how glycans can modulate immunological functions during viral infection. And in particular, I will focus on something called glyco-immune checkpoints. And I'm sure all of you are quite familiar with classic immune negative checkpoints that are work by protein-protein interactions. So for an example, the protein PD-1 expressed in immune cells bind to another protein called PD-L1 on surface of target cells, including cancer cells, and these interactions inhibit immune functions and allow, for an example, cancer cells to evade immune surveillance. Now, and that's why this is obviously a promising ways to enhance immunity during cancer and other diseases. So now, recently it become clear there is another class of immune negative checkpoints that work by binding to sugars, and that's why they call glyco-immune negative checkpoints. So again, let me start this part by telling you that is every immune cell express several sugar binding proteins, or also called lectins, on their cell surface. And the binding between some of those lectins expressed on the immune cells and specific or certain sugar on the surface of target cells plays very important role in the ability of these immune cells to recognize and target these target cells. And that's why many cancer cells, many virally infected cells, try to decorate themselves with specific sugar in order to ensure binding to some of those inhibitory lectins and evade immune surveillance. And obviously, this is representing a good possibility to block those glycoimmune negative checkpoints to enhance immunity. So today I will focus on one of those glycoimmune negative checkpoints, and I will first show you how this glyco-immune negative checkpoints interactions can play an important role in regulating innate immune functions during HIV infection, which is some of this work was published in this paper last year in this project that was led by Samson Adeniji, a postdoc in our lab. And then after this, I will show you how the exact same glyco-immune negative checkpoints could play an important role in modulating immunity against SARS-CoV-2 infection. So start with the first part on HIV. And before I get into how this glyco-immune negative checkpoint can impact HIV immunity or immunity against HIV, I want to spend a quick minute telling you why we still need to understand immunological functions during HIV infection, despite the fact that we currently have a very effective antiretroviral therapy, or also called ART, that can effectively suppress viral replication. And the reason is that antiretroviral therapy, or ART, does not cure HIV. And despite how many years of therapy, there always be a large number of HIV-infected cells that persist in the blood and in multiple tissues in the body. And these cells can hide from the immune system in a ways we don't understand. But because of those cells, regardless how many years in therapy, once stopping it, the virus rebound within a couple of weeks. It doesn't matter if the patient on therapy for 5 years, 10 years, 15 years, once stopping therapy, the virus will rebound very quickly. So a lifelong therapy is necessary for those 37 million people living with HIV worldwide, including 2 million children. And now lifelong therapy is associated with several problems. I will just highlight one of those problems now, which is the fact that as people living with HIV, regardless of viral suppression, are still suffering from high incidence of many aging and inflammation-associated diseases, including neurological impairments, cardiovascular diseases, cancers, and whatnot. Now, these diseases happen at much higher rate and sometimes at much lower age in HIV-infected individuals compared to their HIV-negative counterparts. So that's why there is still a lot of interest in finding a way or ways to enhance immunity, to eliminate these HIV-infected cells, to cure HIV and prevent these significant complications. So now this project started in our lab a couple of years ago when we asked the question, do HIV-infected cells have different glycans on their cell surface compared to HIV-uninfected cells? And taking a long story short, because this was published in this paper a couple of years ago, we indeed found that is the HIV-infected cells have a glycomic signature to them that different than the glycomic signature on the surface of HIV-uninfected cells. And one of the features of this glycomic signature was an upregulation of this sugar called alpha-2,3 sialic acid. And this was interesting for us because alpha-2,3 sialic acid is a sugar ligand of one of those glyco-immune negative checkpoints, lectins, I mentioned to you. And this one in particular called Siglecs. So Siglecs is a sugar binding protein that expresses on the surface of many immune cells, including on a subset of the cytotoxic NK cells. And when Siglecs expressed on NK cells bind to alpha-2,3 sialic acid expressed on target cells, this binding send an inhibitory signal to the NK cells and allow the target cells to evade NK immune surveillance. So now knowing that HIV-infected cells do have a high level of alpha-2,3 sialic acid on their cell surface, we naturally asked the question, do Siglecs sialic acid interactions contribute to the ability of HIV-infected cells to evade NK immune surveillance? But before we even attempt to answer this question, we wanted to do a very simple test. We reasoned that is if these NK cells expressing Siglecs, this inhibitory lectin Siglecs plays any role whatsoever during HIV infection, we should see a correlation in vivo, in patient, between the percentage of these NK cell that express this inhibitory receptor Siglecs here in the x-axis in both the top and the bottom panels, and level of HIV, either level of HIV viremia in the plasma on individual when they are not on therapy, or remnant HIV when the patient are in therapy. And we reason that is we should see a positive correlation, right? The more NK cell that express an inhibitory receptor, the more virus, more immune inhibition, more virus, right? Makes sense. But to our surprise, we found a negative correlation in both cases, which didn't make any sense to us. Why more NK cell that expressing an inhibitory receptor to immune system, to immune function, correlate negatively or with correlate, the more of it correlate with less virus? And at this point, we thought that is maybe just a random in vivo correlation. It's just a correlation after all. It doesn't show any causality, and maybe it's just a random correlation that doesn't mean anything in vivo, and we just decided to ignore it. Until this paper came out where the authors investigate the role of the exact same NK subpopulation that express Siglecs, but now during hepatitis B viral infection. And they also reported this correlation in their paper. Between the NK cell that express Siglecs and level of hepatitis B viral DNA. And unfortunately, the author didn't try to explain this counterintuitive correlation, but it struck us in how similar it is to what we found during HIV. Again, negative correlation. And at this point, we thought that is maybe these negative correlation actually real. Maybe they do have a significant biological meaning. That is, we just don't understand what is this meaning. And it was at this point that it came to us that is maybe we are confusing the subpopulation that express Siglecs with the Siglecs. Now, the Siglecs, the molecule itself is inhibitory. There is definitely inhibitory. There is no question about that. But that doesn't necessarily mean that is the NK subpopulation that express this inhibitory receptor are inhibited and are less cytotoxic. Because the cytotoxicity of NK cells is a balance between the expression of several NK activating receptors and markers and several NK inhibitory receptors and markers. And it's possible that is this NK subpopulation that express Siglecs, regardless of their expression of Siglecs, are actually highly activated and highly cytotoxic, because maybe they do express high level of several NK activating receptors and markers, and maybe low level of other inhibitory receptors and marker regardless of Siglecs expression, which would actually explain our negative correlation here. So we started to explore this alternative hypothesis that is the Siglecs positive NK cells are actually more activated and more cytotoxic against HIV infected cells compared to the Siglecs negative subpopulation. And we started exploring this by simply measuring the expression of several NK activating and inhibitory receptor on the surface of Siglecs positive cells here in solid dot, and comparing them to compare it to the expression on Siglecs negative cells here in open circles. And we use cells isolated from either HIV uninfected individuals, HIV infected individual on therapy, or HIV infected individual not on therapy. And as you can see here, the expression of several NK activating receptors and markers, including CD16, CD38, NKp30, peripherin, DNAM, are higher on Siglecs positive cells compared to Siglecs negative cells. On the other hand, the expression of several important inhibitory receptors to NK, like NKG2A, TIGIT, are lower on Siglecs positive cells compared to Siglecs negative cells, regardless of HIV disease status. So far, we know in vivo that is these cells having an activated phenotype, and they do exhibit a negative correlation with HIV, all suggesting that these cells are actually more activated and more cytotoxic against HIV-infected cells. And if you really think about that, this is not something very weird, because classic immune-negative checkpoints like PD-1 are usually expressed in highly activated, highly differentiated T cells. That is, they now need to express PD-1 to restrain their high activation and cytotoxicity. So now we wanted to explore if indeed Siglecs positive cells are more cytotoxic against HIV-infected cells compared to Siglecs negative, which is we use different approaches in order to answer this question. In one of the approach, we just simply deplete Siglecs positive cells from NK. As you can see here, total NK will have about 40% of the cells expressing Siglec 9. We can nicely deplete these Siglec 9 positive subpopulation, and then we use the whole NK cell that contains Siglec 9 subpopulation and the Siglec 9 depleted NK cells in a killing assays against HIV-infected cell lines, obviously at the same effector to target ratio, with the idea if Siglec 9 positive NK cells plays an important role in killing HIV-infected cells, we should see a reduction in the killing of HIV-infected cells when we use the Siglec 9 depleted NK cells, which is indeed what we see here. You can see when we measure NK degranulation or cytotoxicity using two different assays, LDH release and CFSE red assay, you can see the total NK cells here in blue can degranulate and kill HIV-infected cells better than Siglec 9 depleted NK cells here in yellow. Kind of indirect evidence that is indeed Siglec 9 positive cells are more cytotoxic against HIV-infected cells compared to Siglec 9 negative cells, which is something we want next to directly evaluate by sorting Siglec 9 negative NK cells from Siglec 9 positive NK cells and then co-culture them with HIV-infected cell lines. And as you can see here in the left panel, the Siglec 9 positive NK cells are indeed more cytotoxic compared to the Siglec 9 negative NK cells toward HIV-infected cells. And in the left side, that is confirmed when we look at the degranulation of these cells. And indeed, again, the Siglec 9 positive NK cells can degranulate more toward HIV-infected cells compared to the Siglec 9 negative NK cells. So we know so far that is indeed the Siglec 9 negative positive NK cells are more activated in vivo and more cytotoxic in vitro compared to the Siglec 9 negative NK cells, which is consistent with the negative correlation we see with HIV in vivo. But then the question, Nick, is what about their expression of Siglec 9? Yes, these NK cells expressing Siglec 9 are more cytotoxic against HIV. But is that possible that is their expression of the inhibitory receptor Siglec 9 is still restoring their high cytotoxicity? And if we were to block Siglec 9, we can even unleash a higher cytotoxicity from these cells against HIV-infected cells? And the answer is yes. When we made a blocking antibody for Siglec 9 with help of GeneScript, and we used this blocking antibody in a co-culture assays against HIV-infected cells using total NK cells, we can indeed dramatically enhance the ability of these NK cells to kill HIV-infected cells. And this antibody was specific for Siglec 9 because if we now do this experiment, but instead of using total NK cells, we use a Siglec 9 depleted NK cells, the antibody do not have any effect because there is no target cells for it. This experiment was done using cell lines infected with HIV, but we could also recapitulate this data using autologous assays where we isolate BBMCs from individuals, and we isolate CD4 from those BBMCs, infect them with HIV, and in the same time, we isolate NK from the same donors, and then we co-culture those HIV-infected CD4 T cells, and NK cells in the presence or absence of an isotype control or the Siglec 9 blocking antibody. And then we can do different measurements. We can look for the effect of the Siglec 9 blocking antibody on the NK degranulation. And as you can see here, indeed, the blocking antibody significantly enhance the ability of the NK cells to degranulate against HIV-infected cells. We can look for the amount of HIV-infected cells remaining, and you can see that is the blocking antibody compared to the isotype control can cause a reduction in the level of HIV-infected cells. We can take the culture supernatant from those co-culture and try to infect new cells, and you can see that as the culture supernatant from the co-culture in the presence of the Siglec 9 blocking antibody here in red can lead to less infection compared to the isotype control in gray or the no antibody in blue. So now from this part, we know, or we can summarize, that is, we found that is surprisingly, that is this NK cell that express this inhibitory receptor Siglecs are actually highly activated and highly cytotoxic against virally infected cells. And that is maybe due to their high expression of several NK activating receptors and markers like NKp30, DNAP, and lower expression of other inhibitory receptor like NKG2A compared to the Siglecs negative NK cells. However, we also found that the cell expression of Siglecs is still restraining their full cytotoxicity. And if we were to block this interaction, we could unleash even a higher cytotoxicity against virally infected cells from these already highly cytotoxic NK cells. And the next question for us was, now we know that is a Siglecs sialic acid interaction do play a role in the ability of HIV-infected cells to evade NK immune surveillance. The next question, can we design a selective approach to break the Siglecs sialic acid interaction in order to enhance the susceptibility of HIV-infected cells to NK-mediated clearance? And we reasoned that is we can use this enzyme called sialidase. So sialidase is an enzyme that can cut sialic acid from the surface of cells. It can cut sialic acid, including alpha-2,3 sialic acid. And however, if we were to use sialidase, this will just cut sialic acid from the surface of HIV-infected cells and HIV-uninfected cells. So we borrowed a great idea in the cancer field that was done by Karen Bertozzi and others to conjugate this sialidase to an antibody. And for our case, we reasoned that is if we conjugate sialidase to an HIV-specific antibody, the idea here that is the HIV-specific antibody will steer the sialidase to the surface of HIV-infected cells, cutting sialic acid only from the HIV-infected cells without impacting HIV-uninfected cells. So now cutting the sialic acid from the surface of HIV-infected cells will enhance their susceptibility to NK-mediated clearance without impacting the surface of HIV-uninfected cells. And indeed, when we produce those HIV antibody, again with the help of GeneScript, and then conjugate them to sialidase, which is the work of our colleague Han Zhou in Rice University. And then we tested the ability of this conjugate to remove sialic acid from the surface of HIV-infected cells, but not HIV-uninfected cells. So what we did is we mixed HIV-infected cells here in these FACS plots in the right, and HIV-uninfected cells in the left, and both type of cells were in the same tube. And then we stained them for sialic acid here in the y-axis. And when we treated these cells with a small concentration of the conjugates, and in this case, we use an antibody called NIH4546, which is HIV-specific antibody, and we conjugate them with a sialidase. We use 25 nanomole, and you can see here, even at this lower concentration, the level of sialic acid is reduced dramatically from the surface of HIV-infected cells, but not HIV-uninfected cells. We can increase the concentration of the conjugate, and you can see a nice dose-dependent reduction on the level of sialic acid from the surface of HIV-infected cells, but not from the surface of HIV-uninfected cells. So we know there is some specificity to the conjugate in removing sialic acid only from the surface of HIV-infected cells. And the next obvious question, does that lead to potentiating NK killing against these cells? And we tested that again using different models, and I'm here showing you one example when again we cultured or mixed HIV-uninfected cells here in red, and we dyed them with a red color, or HIV-infected cells, and we dyed them with a green color. And you can see that here with this live imaging staining, or also the number of the cells in these bar graphs. Then we added to those cells NK cells isolated from HIV-infected individuals, and you can see the NK will start killing HIV-infected cells. You can see about twofold reduction in the level of HIV-infected cells compared to the control, with no impact on the HIV-uninfected cells here in red. We can add an isotype control, and nothing much happens. we can add an HIV-specific antibody at 2 different concentration, and you can see the 2-fold reduction in the level of HIV-infected cells became 5 or 6-fold reduction, because those antibody will elicit antibody-mediated cytotoxicity. And you can clearly see that is in the level of HIV-infected cells dramatically reduced. And you wonder if there is even a space for the conjugate to reduce this further. And indeed, this, when we add the same concentration of the conjugate instead of the antibody, you can see the 5-6-fold reduction become 25 to 50-fold reduction, and you can barely see any HIV-infected cells in the culture. And most importantly, with very minimal impact, if there is impact at all, on HIV uninfected cells in red. Again, this experiment was using cell line infected with HIV, and we could recapitulate the same result using autologous system, primary cell, where we infect CD4 T cells and co-culture them with BBMCs from the same individuals in the presence or absence of the antibody or the antibody conjugates. And we can, again, measure 2 different things, either NK degranulation, either as expression of CD107 or co-expression between CD107 and interferon gamma, or CD107 and TNF alpha. And in all of those cases, you can see that there's 2 different concentration of the conjugates here in bright red can lead to higher NK degranulation compared to the exact same 2 concentration of the antibody alone here in dark red. And obviously this resulted in a reduction in the level of HIV-infected cells when using this conjugates again in bright red compared to the antibody alone in dark red. So just to summarize this part, and just as this is just a quick example of how studying glycobiology could allow us to identify a novel glyco-immune checkpoint mechanism that may be exploited by HIV-infected cells, and I will show you how that might be also true for other virally infected cells, to evade immune surveillance, and how this information was actually useful and allowed us to develop a proof of concept approach to selectively enhance the susceptibility of HIV-infected cells to immune-mediated clearance by using this sialidase HIV-specific antibody conjugates, which is now we obviously advancing to animal studies. But that will lead me to the second part of my presentation, which is a question, is this specific for HIV, or this sialic acid-Siglic interaction could also contribute to the ability of other virally infected cells to evade immune surveillance? Which is obviously, I will focus on SARS-CoV-2 infected cells, and the work I will show you next is work of Britt Massini, another postdoc in our lab. And probably all of you know that severe COVID-19 is associated with several alterations to the profile of many immune cells in our body, including alteration to the profile of NK cells. This has been shown in several publications, but it's unclear whether this alteration can cause alteration to the functions of these NK cells. These NK cells are expressing different markers, but did that mean that is the NK functions during severe COVID-19 are impaired? And as all of you know, NK cells can either kill directly by direct cytotoxicity, making a direct synapse between the NK cells and infected cells, or they can bind to the Fc portion of antigen-specific antibody in order to make another synapse that will allow them to kill by something called antibody-dependent cellular cytotoxicity, or ADCC. So it's not clear if these anti-SARS-CoV-2 NK function, either direct cytotoxicity or ADCC, are impaired during severe COVID-19, and what host factor might modulate these functions. So we first wanted to answer this question, whether this alteration lead to reduction in the functions. So we collected samples from SARS-CoV-2 negative controls, individual with mild COVID-19 or hospitalized COVID-19. And first, we confirmed what is known in the literature, that is indeed the NK cells in those individuals with hospitalized COVID-19 have different alteration. They do express lower CD16, higher CD57, higher Siglecs-7, several other alterations. So we know that these individuals we collected are representative of what is going on in the literature. Next, we wanted to ask if these cells, NK cells, have lower functionality. So we did that by either culture these BBMCs from those negative, mild, or hospitalized COVID-19 patients by themselves, or co-culture them with a target cells, a cell line that is expressing SARS-CoV-2 spike protein. We did this co-culture also between the PBMCs and the target cells in the presence or absence of an antibody that is non-specific for SARS-CoV-2 or SARS-CoV-2 specific antibody in order to evaluate ADCC. And then we left this culture for 16 hours, and then we measure NK degranulation as a marker of NK functionality. In order to measure direct cytotoxicity, we just subtracted the NK degranulation we get from the co-culture between the PBMCs and the target cells to the PBMCs alone. In order to measure ADCC, we subtracted the degranulation we're getting when we use an antibody specific for SARS-CoV-2 from the co-culture when we use an antibody non-specific for SARS-CoV-2 after normalizing both conditions to the PBMC's control. So indeed, as we hypothesize, that is, this alteration are indeed associated with less functionality. These NK cells from individuals with hospitalized COVID-19 are less capable of degranulating as measured by the co-expression of CD107 interferon gamma or releasing cytokine like interferon gamma or co-expression of interferon gamma and TNF alpha compared to the NK cells isolated from the mild or negative or SARS-CoV-2 negative individual. And that is in the case of direct cytotoxicity. This is also true for ADCC. These cells are less capable of eliciting ADCC compared when they are isolated from hospitalized COVID-19 patient compared to individual with mild COVID-19 or SARS-CoV-2 negative individuals. We also measure the level of how much virus in the blood in those individuals by measuring the level of N antigen. And as you can see here, in the x-axis, the ability of the NK cells to either elicit direct cytotoxicity and degranulate negatively correlate with how much virus in the body. So the less NK functionality, the more virus. So now we know that is the NK cells from hospitalized COVID-19 patients are indeed less capable of eliciting anti-SARS-CoV-2 direct cytolytic and ADCC activity compared to NK from control. We ask us the question, does sialic acid-cyclic interaction contribute to this less functionality? And as we found during HIV infection, we indeed also found that is the Siglecs positive NK cells here in solid dots are capable of eliciting higher ADCC compared to the Siglecs negative NK cells. So they are also highly cytotoxic against SARS-CoV-2 cells. You can see that when we use cells from negative, mild, hospitalized, that is the Siglecs positive NK cells here in solid dots, capable of eliciting higher ADCC compared to the Siglecs negative NK cells when we measure CD107 degranulation. Same true when we measure cytokine release, interferon gamma, TNF alpha. Again, these cells are capable of degranulating and releasing cytokine better than the Siglecs negative control. Even when we measure also degranulation plus cytokine release, we get the same message. When we then correlate the ability of the Siglecs positive or Siglecs negative cells to elicit either direct cytotoxicity or ADCC and level of virus antigen in the body, we found that there is no, the ability of Siglecs negative and Siglecs positive cells to elicit ADCC do correlate negatively with the N antigen, but without difference between the Siglecs negative and Siglecs positive. But when we focus on ADCC, we found that as the Siglecs positive cells, ability to degranulate or release cytokine correlate negatively with how much antigen only in the Siglecs positive cells, but not in the Siglecs negative cells, suggesting that as the Siglecs positive cells are playing an important role in the ADCC activity against Siglecs. And next, we then also ask us the question, do these cells have an activated and mature phenotype during SARS-CoV-2 as we observed it during HIV? And the answer is yes. We indeed found that as a Siglecs positive NK cells, again here in solid dots, are expressing high level of CD16, which is again important marker for eliciting ADCC compared to Siglecs negative cells. Again, regardless of disease status, these cells are more mature. They are expressing CD57, which is a maturation marker compared to Siglecs negative cells. They do express high level of NKG2C, which is an activating markers. and they do express lower level of NKG2A, which is an inhibitory marker. So similar to what we found during HIV, these cells are more activated and more cytotoxic against HIV and SARS-CoV-2 infected targets compared to Siglec-9 negative cells. And again, the next question, these cells still express Siglec-9, and if we were to block this interaction again using a Siglec-9 blocking antibody, that's again, we use the help of gene scriptomaking, can we enhance the anti-SARS-CoV-2 ADCC activity? And as you can see here, compared to an isotype control, when we use a Siglec-9 blocking antibody, we can see an increase in several markers of degranulation and cytokine release, CD107 co-expression with interferon gamma, or TNF-alpha, or even TNF-alpha expression or co-expression between TNF-alpha and interferon gamma, that is the Siglec-9 blocking antibody, could enhance the ability of those cells to degranulate. But the next question is this degranulation is actually equal to lysing the cells. So we use this targeting killing assay when we have a target cells expressing SARS-CoV-2 spike protein, and these cells have one part of luciferase, and the media have the other part of the luciferase. So we co-culture this with NK cells in the presence of anti-SARS-CoV-2 spike antibodies. And the idea is when the cells are being lysed, the part of the luciferase inside the cells will go to the outside the cells, bind with the other part, and we can measure the cell lysis by luciferase activation. And the idea here is when we do that without Siglec-9 blocking antibody, we see some level of killing. But the hypothesis, if we were to block Siglec-9 using the Siglec-9 blocking antibody, we can even lead to higher cell lysis, that is, we can measure by luciferase. So we examine if that is true, and the answer is absolutely, this is true. When we use an anti-Siglec-9 blocking antibody, we can enhance the lysis of the SARS-CoV-2 specific target cells by ADCC. So again, going back to this model, similar to what I showed you with HIV, in this model, we also found that the Siglec-9 positive cells are indeed highly activated and highly cytotoxic against SARS-CoV-2. So it's not just specific for HIV and can be applicable for many other viral infections. And that is, again, probably due to their high expression of several NK activating markers and receptor like NKG2C, and lower expression of inhibitory receptor like NKG2A compared to Siglec-9 negative NK cells. However, again, we found that the Siglec-9 sialic acid interaction is still restraining this cytotoxicity. And if we were to use a blocking antibody for Siglec-9, we can enhance the NK cytotoxicity against SARS-CoV-2 infected cells. So this is just one quick example, actually, with how glycans could be important for immunity against HIV, or virally infected cells. But again, in the last few years, there is many advances in this field of glycobiology. And there is many areas that is, in these advances, we can learn more about viral host interactions. And if you want to learn more about that, I will refer you to the several review article and several paper we write in several direction, that is, we can learn from the recent advances of glycobiology in order to better understand viral host interaction against HIV, against SARS-CoV-2, and perhaps against many other viral infections. And with that, I would like to go to my acknowledgement slides. First and foremost, thanking all of the volunteer participants in our different studies, thanking different folks in our lab who did the work, our collaborator with and without outside Western, our funders, and thank you for your attention, and I will be happy to take any questions.
And thank you, Mohamed. We appreciate that presentation. And would you do us a favor and go ahead and refresh your screen at the top left, because you were not, you were frozen during the part of your presentation. And while you are refreshing, I just want to welcome our audience to the Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Mohamed, it looks like we already have some great questions coming in, and thank you so much for refreshing. Our first question is, since In addition to Siglecs, are there any other glycoimmune checkpoints that had been reported with antiviral or anti-tumor immunity?
Yeah, definitely for anti-tumor, as I mentioned, there is a lot of great work is being done in this area for in the cancer field. So there's many other lectins that could play a very important role during cancers, including selectins, which is bind to specific kind of cause. There is also DC-cyan, galactins are very important immunomodulatory lectins. And again, there is many different papers showing that is many of those lectin and their interactions could negatively or positively impact immunity in different ways, in a way that is can impact cancer, but they also can impact virally infected cells. We have also published paper on galactins. We have working on selectins, but as I mentioned, this is like an non-necessary, very mainstream science. There is an evolving field rapidly, and I think we will see even more and more in the next few years. And the important role of glycan that is usually neglected in modulating immunity during both cancer and infectious diseases.
Thank you so much. Now, since anti-tumor immunity and antiviral immunity are two different checking scenarios, do you develop the antibody specific to each scenario or just one?
Well, I think the important part, because those glycans are very abundant. They are in every cell in our body. So the main challenge is to how to get specificity. And that's why we, for an example, in our example on HIV, we wanted to conjugate sialidase to HIV specific antibody to ensure that is we are interrupting this interaction only on the surface of HIV infected cells, because sialic acid, Siglec are important for immunity. So if you were to block them non-specifically, you might get non-specific reaction like inflammation, autoimmunity. So I think it will be very important to try to be adding a specificity to your antibody, either by making them bispecific, trispecific antibody, that is, they go and target only your virally infected cells or your cancer cells without impacting your healthy cells. So if that will be the goal, you will probably your the Siglic antibody part will be the same, but you will need to add something to it to infer a specificity, which is will be obviously different between tumor and virus and between different viruses.
Thank you, Mohamed. And I want to thank our audience for these great questions. Our next question: have you confirmed that Siglecs 9 ligands are expressed on SARS-CoV-2 or infected cells?
Yes, yes. So do the not from we confirmed that using these cells we are using in our assays, but we did not do that using actual cells infected. We didn't have an access to that, like real cells infected. That's the cells we infect in vitro, or we or do express a SARS-CoV-2 spike, and we use them in our killing assays. Those cells do have the Siglec 9 ligand. But obviously, it will be important to know if the actual cells in the body, the lung cells, that do express, are naturally infected with SARS-CoV-2, do express alpha-2,3 sialic acid. Many cells, most cells in the body will express alpha-2,3 sialic acid. It's not something specific to virally infected cells, but the question will be, do they express it more? We could find that during our HIV studies, because obviously we can isolate easily T cells from the blood and examine their glycome. But obviously for SARS-CoV-2, that will be a much challenging task, because we will need cells from the lung, that is, we know, infected with SARS-CoV-2 and whatnot, which is something we haven't done.
Thank you, Mohamed. And how do you connect those Siglec checkpoints from anti-tumor immunity to antiviral immunity?
Yeah, that's an interesting question. So in our studies, we keep being surprised in how viruses seems to employ same or very similar ways to evade immune surveillance, similar to cancer. So in the cancer field, it is known for a long time that is cancer cells have high level of sialic acid on their cell surface, or something called hypersialylation. They do intentionally express high level of sialic acid in order to ensure binding to Siglec and evade immune surveillance. So we were surprised that as many viral infection do the same thing. They do express, when they infect the cells, they find a way to increase the level of sialic acid on their cell surface in order to again, maybe evade immune surveillance. Many cancer cells do express the ligand of another important lectin called selectin, because that helps them to disseminate, to move from the blood to the tissues, which is important for the cancer cells to metastasize. We found also that as virally infected cells can enhance the ligand of selectin, which is called sialic X, which helps cells to migrate. So obviously, we don't have a very clear evidence, but from our different studies, we keep finding that there might be a very shared mechanism of how both cancer and virally infected cells take advantage of the regular cell biology, because sialic acid Siglec interaction is not there for cancer to evade immune surveillance or for virally infected cells to evade immune surveillance. It does have a very important role in controlling inflammation and autoimmunity in the body. Selectin interaction are not there for, again, virally infected cells or tumor to disseminate between tissues. They are there to help immune cells to move to the site of inflammation. But somehow both cancer cells and virally infected cells could hijack those systems for their benefit. And again, we keep finding that both tumor and virally infected cells seems to use shared mechanism, and that's why we sometimes, learning from the cancer field and apply to virally infected cells and vice versa, because of this potential shared mechanism.
Thank you so much. And what type of carbohydrate recognize the lectin, and what lectin do you use?
Yeah, it depends on the lectins. Our body have many lectins, tens of lectins, and each one of those lectins do have a specific glycan binding specificity. And it's not 100% clear, actually, but we know, for an example, as I mentioned, Siglecs usually bind to sialic acid. There are many Siglecs. I think it's up to 16 Siglecs, and they are different between mice and human. Each one of those Siglecs can bind to a specific kind of sialic acid, because not all sialic acid are the same. Selectin, there are 3 of them: B-selectin, E-selectin, L-selectin, and again, they bind to specific kind of fucosylated glycans and so on and so off. So, the different lectin do have different glycan or carbohydrate recognition, and those are the ones in human. Mice will have different lectin with different glycan recognition. Plants will have their own lectin with lectin recognition, and actually the plant lectin are very commonly used in the field of glycobiology in order to measure glycans, because their glycan specificity is relatively well understood. We use different lectins in our study, depends on the project and the goal.
Thank you so much. Now, we have time for a couple more questions. How specific is sialidase in eliminating the sialic acid ligand that Sialic 9 recognizes? And this is a 2-part question. What can we find where can we find alpha 2,3 sialic acids in our body and our tissues, or any particular cell types?
Yeah, so the sialidase will not be specific to the Siglec 9 ligand in particular. Sialidase will just remove Siglec 9 from sialic acid for all sialic acid. When we test it, it does take all alpha-2,3 sialic acid, but it will also take the sialic acid from other ligand. And that's why it was very important to conjugate that to HIV-specific antibody, because if we wouldn't do that, you would just remove all sialic acid from HIV uninfected cells. We reason that is removing all sialic acid from HIV-infected cells, from the surface of HIV-infected cells, might be good, might be even better than try to target alpha-2,3 sialic acid in particular, because there are other Siglecs, like Siglec 7, also can help these cells to evade NK immune surveillance. By using sialidase, you will remove the ligand of Siglec 9, Siglec 7, other Siglec, which is again will not be something recommended if you will not conjugate these sialidase to the HIV specific antibody, because again, doing that, you will lose your body a lot of inflammatory checkpoint. You might cause inflammation. But when we use that, when we conjugate that to HIV specific antibody, removing all kind of sialic acid from HIV infected cells could be good, because it can initiate multiple immune cells against these cells, but the sialidase will not be specific. And for the second part, where we can find alpha-2,3 sialic acid, is everywhere. Everywhere in the body will have alpha-2,3 sialic acid. It's abundant. Our study just started by the fact that HIV infected cells have higher level of it, but many places, many cells will have alpha-2,3 sialic acid. Usually in the body, one of the most areas that have a lot of sialic acid, a lot of sugars in general, are the gut and the gut epithelial cells, because these glycans plays a very important role in communication with the microbiome. The microbiome like to cut this sugar and eat it. And so you will find the gut, the lung are heavily glycosylated. Probably the gut are the most glycosylated area in the body. But as I mentioned earlier in the presentation, every single cell on Earth, without exclusion, there are some cells that do not have nucleus, but they will still have a glycome, just showing you how important having a glycan is as an kind of like an essence of living. But obviously different cells will have more or less of different kind of sialic acid, and I'm not sure, to the best of my knowledge, that is categorized yet.
Thank you so much. Now, we have time for one more question. Are Siglecs-7 ligands also expressed on HIV-infected cells, as Siglecs-7 is an inhibitory receptor on NK cells, and Siglecs-7 expression correlates with other activation receptors?
Yeah, that's a great question. So indeed, Siglecs-7 ligand will be expressed on HIV-infected cells. Again, it's not 100% clear the ligand of each Siglec. It's believed that is Siglecs-9 bind to alpha-2,3 sialic acid, while Siglecs-7 can bind to alpha-2,8. Some people say alpha-2,6. And obviously alpha-2,8 and alpha-2,6 will be expressed on regular cells as well in HIV-infected cells. We have looked at Siglecs-7, and we didn't find that is the Siglecs-7, the percentage of Siglecs-7 expressing cells correlate with HIV as the Siglecs-9 positive cells do correlate. But however, this was hard because Siglecs-7 expressed in many NK cells. Almost all CD56 DIM NK cells will express Siglecs-7, while only in a population of those will express Siglecs-9. So all of the Siglecs-9 will also express Siglecs-7. But we did some analysis to show that it was the Siglecs-9 more than the Siglecs-7 contribute to the HIV-infected cells to evade immune surveillance. Now, this is for HIV-infected cells. When we use cancer cell line, that is quite different, where the Siglecs-7, but not the Siglecs-9, is more important to the cancer. So everything I showed to you is more about viral infection. And as I mentioned, while there are similarities between cancer cells and virally infected cells, there is obviously also a very clear differences. And that is one of the differences. We found that the Siglecs-7 to be more important against cancer cells, while Siglecs-9 seems to, in our analysis in both HIV and SARS-CoV-2, that is maybe playing a more important role during viral infection. But again, that is need to be more or better studied to be certain.
Mohamed, I want to thank you for your presentation today. Would you like to provide any closing remarks to our audience before we go?
No, as I mentioned, this is just what I presented is just one quick example of how glycan and glycan-lectin interaction could contribute to viral infection. But I do encourage people to just look for our recent reviews and papers, because we study different angles on how, as I mentioned, the recent advances in the field of glycobiology, which is like emerging field, can allow us to better understand viral host interaction. And while in this reviews and in this paper we use either HIV or SARS-CoV-2, or we focus on HIV or SARS-CoV-2, many of the ideas and the concept we discuss in those reviews and paper, I believe could be applicable to many other viral infection. And I think it will be a great opportunity for the viral field to take advantage of these recent advances in this field of glycobiology. Thank you.
Well, I want to thank you again, Mohamed, for your time today and for your important research. And before we go, I'd like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions and questions that were submitted during the live broadcast and those submitted during the on-demand period that were not addressed will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.
Thanks.